Welcome to the tutorial section. In this video, I will use an example to briefly introduce how to use EMPG to search and visualize Chip-seq data. Basically, EMPG provides two ways to search a database. If you would like to know what kinds of regulators directly bind your interest gene, you can use target locus mode. Or if you are interested in the genome-wide binding pattern of specific transcript factor or histone modification, you can use in acid protein mode. Here I'm interested in the epigenetic regulation of a flowering repressor called the flower locus C, or say FLC in Arabidopsis. So we can keep the searching mode and species the same and type in FLC. You also can type in the AGI locus identifiers here. Then search to go to the FLC locus in the Genome browser. Then after find the FLC locus, we can go to select the Chip-seq assay we want. Click the select tracks. Here the list all the Chip-seq assays in current database. It provides information like protein name, antibody, whether it replicates, what kind of genotype, genetic background, and also what kind of tissue are used for generate data. Also, you can click, if the work is published, you can click the PubMed link to find the articles. Okay, we want to find H3 lysine 27 trimethylation at the FLC locus. So I can type in H3 K27 trimethylation. As we typing, the list will be changing, and only the Chip-seq assay related to H3 K27 trimethylation will be kept here. We can see that more than 100 Chip-seq assay related to this histone modification. To quickly find the data sets we want, we can go to the category panel to find the genotype one. Okay. In the genotype one, it lists all the different genetic background material has been used for examining H3 K27 trimethylation in our current database. So we can search the curly-leaf and swing double mutant. These two enzymes actually is two methyl transfers which catalyze H3 K27 trimethylation in Arabidopsis. So we can choose one of them, and also can including one single mutant of curly leaf here. Then we want to compare the level in wild type condition. We can choose wild type. 14 days seedlings. Then we come back to the browser. As we can see, this is the Chip-seq signal from 3 different chip data sets at the FLC locus. We can change the order, make it more customized. We can put the coordinate at the bottom, and put the wild type on the top, and double mutant at the end. Okay, and we can zoom out a little bit to look at the surrounding signals. You also can change the locus by type the gene name or locus position here. If the track height also can be changed. You can click the view here and resize the track. Currently is using 100 pixels. If you want to change, you can change the number. If you want to add more tracks, you can click the select tracks back to the track selector. For example, I can add one more like ring1 A and B double mutant. This is the subunit of PRC1 complex. Here we didn't find it is because in the wild type sample they have more than 47 data sets, so it's maybe changed on the button, so we can cancel the selection for wild type. Now we can see the ring 1 A and B mutant genotype has been shows on the top, so we can click here to add this track. The newly added one will be on the bottom, so we can move this one to the middle. Okay, this is the Kin7 signals at FLC locus. So we can see the wild type that have a high level of Kin7 triamethylation, and in the single mutant we can see dramatic decrease of signals. In the double mutant, even the signals at the 3' end also decreased. However, in the PRC1 mutant, there's no big difference. If you feel this view is interesting, you can click the share button to create a link to send to your colleague or friends. By using this link, they can find the exact same view as you saw. Finally, the browser will remember your current track selection, and it will automatically load all the tracks when you revisit the browser. As loading data and genome information will increase the loading time, it's recommended to close all the selections when you leave the website. So we can cancel all the selections. If you want to go back to your previous selections, you can go to the selector panel, category panel, here using recent used one. It will be listed here. Okay, that's all. If you have any questions and suggestions to this video and our website, please contact us. Enjoy.